right, ichnology is the study of organism behavior and its products. We call those products trace fossils. An expanded kind of definition we've given them is a trace fossil is a product of an organism that's interacting with a medium, and that's not a clairvoyant. You go pay $5 and they read your palm. Some people would call that substrate, but I think it's better to use this term. In an environment that generates a three-dimensional physical structure. So these are highlighted because I'm going to quickly define those. The organism is actually, we have burrowing and boring examples of biota in each of these groups, from the archaea, bacteria, and eukaryota. So trace fossils are scalar. These traces are scalar. The environments then therefore are scalar. They're the surfaces and the micron surfaces of grains to the grains themselves and the sediments in which they're in. So that's the medium, and then the environments, marine, continental, fluvial, lacustrine, lakes, rivers, floodplains, alluvial fans and deserts. And the continental realm is pretty special because marine realm, it's enveloped in seawater. And that, believe it or not, environment may not be as constant as you think it is, but it's a lot more constant than when you're in the continental realm, where you have an unsaturated zone, a saturated zone, and temperature fluctuations. Summertime temperatures, what, 100, 110, depending where you are, or even higher. In winter, we had some brutal winters, minus 30, minus 40 up in Minnesota, minus, what, 50? So you take those, that's pretty big fluctuations. All right, and then also on this small scale, these are borings in volcanic glass underneath the airport of Hilo, Hawaii, 2 kilometers below the surface. That's 40 micron scale. All right, so they etch or bore their way through the glass. Nobody knows why. These are modern iron stromatolites out of Indiana from acid mine drainage. So nice, big, beautiful green mat on the top. The pH of the water is 2. Yeah, I mean, I got jeans that have holes in them. I mean, I could probably sell them for $200 now. They look really stylish. But there's, I mean, the nasty environment. And then B cells. These are millimeters of centimeter scale. Roots, dinosaur trackways from Croatia. Even the nest of dung beetles, Eocene in age, and you can see some of the dung balls still in place where the balls have weathered out. Borings in dinosaur bone. This thing is about 3-4 millimeters in diameter and about 2-3 millimeters in depth. And this is a fossilized part of a subterranean termite nest. That lens cap there is 6 centimeters in diameter as a grad student in the background. These are the things that can reach cubic kilometers. So you can go online and type in, go to YouTube and type in ant nest. And on a NOVA program, they poured in like 10,000 gallons of concrete for like 3 days straight and made a cast of this nest. They didn't know what was going on. And they excavated just a couple acres of it, and you can see it going off away from that. So these things are gargantuan structures that make it into the rock record. So how are they proxies for body fossils? Well, these things, we can even refer to them all as soil biota. The burrows, the traces they make, tell us about the body size, the type of activity, what they're doing, what their effects are on the soil body or on the sediment, and tells us about the amount of time, their presence in the sediment or in the soil. All right, so these are excellent proxies for the organisms themselves. Here's a couple examples. This is one of these May beetles. These are things that tear up your lawn. Golf courses hate them. It makes a burrow. It basically digs in the front, packs in the back. So it's a closed burrow system. Here in this setup, 40 centimeters by 40 centimeters by 2 centimeters deep, there were 3 of these beetles. Chewed it all up in 2 weeks. These are crawfish in Kansas. Water table shallow. It's about 50 centimeters in depth. This burrow is 2.5 meters down to the water table. Same crawfish. All right, so the burrows mark the depth of the water table. This is a barty grub. That thing's going to pupate into a moth. Looks pretty nasty. It makes a lime burrow, but it's lime because when it comes out of moth, it doesn't have really good things to dig with. But these things are great for fishing in Australia, but it leaves an open tube. And then other organisms work together, and this is a small termite mound. And they cooperate and build these structures above and below ground. And here's where we just took a shovel and popped this open and didn't see any termites. Well, that's because I destroyed their home and they're all sort of huddled over here. So I made their 6 o'clock news, destroyed their place. So these structures could represent the work of one or multiple organisms that reflect the rapid amount of activity, the depth of the water table, the different kinds of lifestyles, or the permanent underground lifestyle. So what the trace fossils, the burrows and tracks and trails tell us, these things are basically paleoenvironmental indicators. So in the marine realm, the tetrahedron of indicators or factors, they tell us about the rate of sediment accumulation, depositional energy was high energy, low energy, amount of oxygenation in the water and in the bottom sediment. And then this last corner, it could tell us about the interactions with the medium or the substrate, the salinity, nutrients, hydrology, turbidity, temperature. The continental settings, above and below ground, you can see the tetrahedron are very similar. Big difference is it tells us about the groundwater profile and its fluctuations through 1 year or through multiple seasons. All right, and then what's important about the continental setting is that that's where soil formation takes place. All right, and that subaerial exposure is affected by not only the sedimentary environment that it's in and the hydroclimate, but the amount of mixing by animals and by separate physical processes, and of course those soil forming factors, chlorine, climate, organisms, relief, parent material, and time. All right, so pretty strong factors. And why should we care? Well, we worry today about our critical zone. The National Research Council, instead of talking about soils, the soils are like, this is actually really important. This is the critical zone of the earth. So from the top of the canopy of vegetation down to the groundwater table, the phreatic zone, that's the critical zone where these complex interactions that are physical, chemical, and biological take place and transform not only the biomass, but the physical material into soils. And soils are important because, well, we're in farm country here. This is our food. It also regulates runoff and infiltration of sediments, transfer of nutrients in terrestrial life forms and back again, and storage and sequestration of carbon dioxide, greenhouse gases. So if we don't take care of our critical zone in our soils, well, we're in trouble. Well, how do you understand trouble? Well, you can go out and look for it. Then you get a better education. Or you can go into the geologic record, where it already happened, and look to see how the critical zone has been affected through time from environmental perturbations, climatic perturbations, and if we're lucky, a big extraterrestrial impact. So you get extraterrestrial perturbations. All right? So the rest of the talk, I'm going to take us through what we've seen in the Permian and in the Triassic deposits, and show you the traces, show you what we think they mean, and put it all together, and tell us about what they say about the mass extinctions and the climatic setting and some of the evolutionary significance. So you can wrap it up from there. All right, so this is our view of Lampion Peak, and it's in this area here. The sediments record the variation of large floodplains and floodplain lakes and smaller graded river channels with lots of trees and in situ stumps. So we're going to take a look at the lake deposits first. And what we have are a lot of thinly bedded deposits, lots of self-looping, self-crossing trails, very thin, centimeter scale. Here we have look like anabranching scale patterns, anabranching burrow networks. So they're shallow, very shallow feeding patterns made by a variety of invertebrates. Here, the whole surface is covered, but if you look at the sediments directly below that, they're undisturbed, so they don't penetrate very deeply. They're all very shallow. And what's really interesting here, I always put this in here, but there is, see, it doesn't show up really good, but underneath this arrow, there's an impression of an insect. So depending on the lighting and the mood of the crowd and me, and I don't see it this time, but it's there. There's actually a small body impression of some odd insect, and still don't know what it is. So what we have is this record of life in these lakes, but no body fossils. Then we have these larger patterns, these large grooves, look like parallel tracks. And then you can see them here. Can you guys see these pairs sort of coming together? Here and here. You see this track goes right here like this, down across here like that. It looks like a train. And then when there's another one comes off of here, there's another set this way. And all the lens caps you see, by the way, are 6 centimeters in diameter. So if you see any smaller bits of them, then you know they're like a couple millimeters. And then here you have some impression with some marks here. We've interpreted these as basically the trails of fish. This is a trace fossil called Untitled. So these record the pectoral fins. So basically the thin fins would be the ray fin fishes, and the lobe fin fishes, what are those, sarcopterids? You know, so things like lungfish, coelacanths, those sorts of things. Now how do we, oh, and then these again, trails made by the fins. How do you think we know that? Well, you spend some money, you buy some scuba diving equipment, and you go to Africa. This is Lake Tanganyika, and go diving and watch what the fish do and see how they behave. And you can actually see them making these traces. All right, and you can you see the pairs in here? A little bit harder here, but you can see them here. Those paired markings. Same exact pattern. So we don't have any body fossils. I don't think there's any body fossils of fish, are there? From Beardmore?
There's there's a tooth from coelacanth.
So we got a tooth. Now we got something exactly like
Collected in 1970, actually.
Yeah, they actually thought it was one of their own, right? Thought, it's coelacanth tooth.
Way before I got there.
So the impression, so lobe fin, ray fin fishes, based on this modern comparative work, is that comparative anatomy. All right, so we have more evidence of fish that these lakes were shallow. They may have been accumulating sediment really rapidly, so you don't have deep penetration. Or like a lot of shallow, warm lakes, that the bottom sediments themselves could be anoxic, so you don't get lots of penetrative organism activity. All right, so there's some good examples. Now let's look at the intervals where we start getting a variety of logs and these odd patterns on the fossilized wood. So those lake sediments, those really thin-bedded layers that would have the fish and the insect traces are here. Those standing stumps come from several layers here. So this is basically like a swamp, hardwood swamp forest, but there were hardwoods that are pines, so softwood forests. And so these are basically the tree trunks seen in cross sections. So there's the trunk and the lateral roots. And you can see these in plan view. You can walk over this ancient landscape that's exposed. And there's a variety. There's a lot of black material, manganese oxide. Some of it is carbonaceous. And then even some of the preservation actually has permineralized wood with rings in it. So it's really, really odd. Well, upon closer inspection of a lot of this, these standing stumps and actually fallen over trees, we look carefully, and there's a lot of this cubic fracture pattern to it, and it actually appears like it has been charred. You guys spend time around any campfires? Or you have fireplaces and tend the wood? I mean, there's a very distinct pattern and distinct coloration that we see on a lot of the logs and a lot of the stumps. So I do a lot of work down in Australia, and this is a Carnarvon Gorge. They got a lot of upright and fallen trees that have gone through forest fires. You see the same patterns and cubic fracture pattern that would have represented the outside and also the inside, so they burn off all the bark so you get the inside part of the tree being charred like this. So this has led us to develop a hypothesis that, well, figure it's the Arctic or the Antarctic, and this is at 75 degrees, 70 degrees south latitude. Why would you have forest fires? If you go to Alaska and people up there are like, why do we have forest fires? They know why they have forest fires. They have forest fires there because during the dry season things ain't so wet, you get lightning, and they burn. And the only reason why we spend money so we don't let it burn is because there's a lot of nice property up there. People got houses with national parks up there. You want those to burn. We should just let them burn. Because all this non-burn practice, you saw what happened at Yellowstone. Control burn, and then it was out of control, and you know, you get reindeer running around on fire. Doesn't look good. So what happened to Yukon Charlie, this is what happened when we were up there, things will burn and they keep burning until it rains or it runs out of stuff to burn, and afterwards it looks like this. And then it all falls over, and this is actually in a swamp. In the wet years, you're walking through here and you're up to your knees. So what we postulate is, you know what? There was probably seasonal forest fires burning in those Antarctic summers. Started by thunderstorms, 24 hours daylight, lots of convection, heating. Lightning starts them and they keep burning until they're extinguished by rain or they run out of things to burn. Some people have postulated by the end of the Permian they didn't have a lot of oxygen in the atmosphere. One of our good friends, a famous scientist, said that's why a lot of these things are barrel-shaped, because they're in hypoxia. They couldn't get enough oxygen to breathe, so you get bigger chests and more robust. And they're barrel-shaped though because they're like cows. The Permian-Triassic cows eat vegetation. So that's why they're more barrel-shaped. It had nothing to do with oxygen. Lots of oxygen in the atmosphere, you can have forest fires. Get evidence of that. And what we see in those 1.5 skyscrapers of section is possible increase in fire frequency up section closer to the boundary. So we go up, we see more and more of this evidence. All right, so that's what we have learned from our work in the Fremont. Forest fires, even though there's swamps and lakes and floodplains. All right, now if we go look at examples of the free malformation, and there's an example from Wyckoff Glacier. Instead of being dark sediments or light sediments and highly laminated, you get thick intervals of sandstones that represent braided rivers, and then you get intervals of basically, we think they're mudstone, but it's more like a muddy sandstone. Green color represents floodplains. And you get up close onto this material, and it represents braided rivers and abandoned channels. And a lot of these have fantastic burrows in them. And these burrows here represent the trace fossil cylindricum. There's some sort of arthropod burrow. Well, when you do comparisons with modern burrowing organisms and you look at modern rivers, meandering and braided, just like these things are, the braided, you see when you get these vertical burrows is when the water level has dropped. So these aren't aquatic suspension feeding organisms. They're probably more like these tiger beetles. Make these vertical burrows. You guys know what tiger beetles are? When they're adults, they're green and they're running across the surface. They can be black. They're really good. You don't kill them because they're great ferocious predators and eat everything else. But what's really cool about these, there's their head at the top of the burrow, and they wait. It's ambush predation, so it's not suspension feeding. Something comes by, they grab it, pull it down in the bottom of the burrow, and rip it to shreds. Sort of like tax time, April 15th. Right? So this represents probably this kind of behavior, and maybe this kind of family of beetle. So not aquatic things, but look at the bartubation there. Just huge abundance. And so that represents stable surfaces. They're just burrowing, reburrowing, reburrowing. Water comes up, deposit new bar, do it again over and over and over. All right, so that's pretty cool. So next time you go out, keep your eyes open for these. Okay, so in this area, we're going to take a look at next. These are abandoned channel deposits. Really, it's finer grain, quiet energy. Think, okay, what are we going to see in there? Well, we see a variety of traces I'm going to walk you through. One trace is called Diplicnites, and there's a couple of varieties of it. The other trace is called Cocklignus, and then we've got some freshwater algae. This kind of Diplicnites, you can see the trail here, possibly made by centipedes. See the footprints here, set right over here. And there's another variety, probably made by millipedes, much more complex leg, feet fall patterns through here. So basically, as it's moving through, and as the feet change, it actually, so the front of the critter is making a new set of tracks, and then the back half of the critter is still making tracks from the rest of its body. So all this is going on, it's in an abandoned channel deposit. So these probably weren't underwater, they're just probably thin film of water when the traces were made. So you have water in it, deposit the mud, water levels falls basically at the sediment surface interface, air interface, and they run across it. And then when you have really thin films of water, annelids and nematodes make the trace called Cocklignus, named for the cochlea in the ear. Methodist minister Hitchcock is actually America's probably most famous trace fossil worker. 1853, putting out this stuff, before there's ever really a science called acknowledged. He's also the guy who described giant bird footprints. Dinosaur tracks. Connecticut Valley stuff. All right, so these are aquatic nematodes. You can see all these sinusoidal traces, the surface covered, and with experiments done by multiple people, the only way this happens, there's got to be basically a thin film of water about the depth of the height of the body. So we know it's just a thin film of water. Freshwater algae that was growing at the time leaves the impressions. But one of the most spectacular things that we found there were these traces. So the free mound with braided rivers inland, like Nebraska and Kansas, fresh water, no ocean nearby. These are trackways of horseshoe crabs. All right? So let's put it in perspective. 75 degrees, 70 degrees, south latitude, pretty much where it is now, that part of Antarctica. Rivers and forests and lakes with freshwater horseshoe crabs. In rivers and in lakes. All right, so these tracks are identical to their marine tracks. There are no marine realm critters. You can see the feet patterns here. This is even better. Take a look at the pattern of these, and we'll show you a couple slides. So this is the underside of a bed. This is the top side of the bed. And from the tracks that have been described from these horseshoe crabs, modern and ancient and marine settings, they're identical. And you can see that pattern, this track pattern right here. Same pattern. These are from off of Delaware Beach, the spring mating season, or spring is actually early summer mating season. Leaving all sorts of trails and head capsule marks. These are examples from the Upper Triassic Chinle Formation, Colorado Plateau. Same sorts of patterns. These are freshwater as well. But these are Upper Triassic or Late Triassic, 200, 210. These from the Fremont, and from Action, we also have them in the Buckley. They're 250, 251, 249, much older. So we know that they're in the equatorial regions at the time, and they're down at the poles. All right, and then here's a number of examples also from the Chinle, and from, can everyone see the horseshoe crab there? This is from the Morrison. Who cares about the sauropods? Yeah, allosaur, big teeth. They're horseshoe crab. Do we have any horseshoe crabs in freshwater today? No. So they made it into freshwater, did good, and then went extinct. But they're still in the oceans. So what does all this tell us? Well, the oldest undisputed freshwater horseshoe crab trackways, maybe Pennsylvania. We got things in the Antarctic, at Permian and Triassic. People still argue about these. There's no argument about these. So this evidence, finding them in Antarctica, tells us that perhaps maybe during the assembly of Pangea, from the start of the Pennsylvanian into basically the Permian, forced these things or helped encourage them to move landward or go extinct, or as they're being trapped, they evolved to be able to move into fresh water. So you take all those shorelines of all those continents and squeeze them together. You go extinct, you adapt or you go extinct. So if we take a look at the history of horseshoe crabs, they evolved down here in the Cambrian, they're marine, they start moving into estuarine systems in the Pennsylvanian. The best evidence now that we have comes out of Antarctica. So we got them in freshwater, definitely for sure, in the late Permian into the Triassic, both hemispheres, for sure, and maybe even down into here. And the last freshwater known freshwater horseshoe crab trackways and impressions we have are from the Morrison. And that's the upper Jurassic, late Jurassic, and then the rest are marine. But what's important about that is, what's it tell us about temperature? Well, if you look today where they're distributed, they're pretty much temperate, down into the subtropics, down into the equatorial regions. So these things don't get frozen in and don't live in environments where the water freezes. All right, so these things tell us that those aquatic systems were much more equitable than we actually thought. All right, so Antarctica at that time wasn't cold and miserable. It was probably more like, you know, the climate south of here, or about here, perhaps. All right. So we look elsewhere, and we have lots of trace fossils. Something called fur sickness, haploticnus, Aernicolites, and more Undicna. All right. So our Aernicolites paired U-shaped tubes. There are the tubes there. And they're similar to the tubes made by modern mayflies. And these things are basically suspension feeding organisms, and they basically are filter feeding, and they filter water through their burrows. And we know that we've actually got these out of Clinton Lake in Kansas. The water level's low and the burrows are exposed. We can go out there, collect them, pop them open, and there they are. There is our Undicna, so our fish trails again. So these are in the free mouth. So we still have our fish. We have our, this is the lobe fin fish, the Sarcopterygians. Maybe it's a coelacanth. And then we have other kinds of mayfly feeding trails here. And then this thing, haploticnus, is probably made by midgefly larvae, things like chironomids. Live in the bottom of lakes and rivers and streams and any little stinky, nasty pool of water. And these are the non-biting ones, so this is good. It's the other ones that are bad. All right, so we have all this aquatic life that's supporting the large aquatic life that may be supporting things that are eating them. So we look in these same deposits, and we have a variety of footprints that we found. And half of these things I found while I was on the phone with my daughter. Calling in to check in, see what was doing, see the sat phones, and you know, walking and talking, like, hey, I just found some footprints. So, walking through some of these, this is a thoracic track, and if you see there's basically the paw and the claws, the toes, 1, 2, 3, 4, preserved over here, probably of a Lystrosaurus. And the trace fossil name would be Brasilignium. So these things weren't probably walking underwater. You can actually see the root traces around this thing. So it's basically the water level falling. This is rippled surface. Plants are growing on it. It's still a little soft. And they're walking through it, leaving their footprints. Can you see the footprint here? Like a nice handprint. Thumb, claw, toe, toe, toe. The nails on the tips. And bit one over here. This is an archosaur track. The trace fossil name is Rhynchosauroides. It's some sort of reptilian footprint. It probably represents an archosauromorph. And the possible trace maker is Prolacerta. Prolacerta. I'm Greek. I can't pronounce Latin. They should make these more Greek. So Prolacerta, and there's actually fossils of it found in Antarctica. Right? But we don't have any body fossils here. We're those fossils found in Shackleton, right?
Yeah.
Yeah, so there isn't any of those things here. So those traces are telling us, oh, look, we got these guys walking around, and it's small, really small. And then we had things, we had Phil Curry out with this. We found this, can you see this nice 3-toe track? Equidimensional, 1 toe, 2 toe, 3 toe, back here to the heel. Phil told me, if this was Cretaceous, I could tell you what it is. Never expect to see anything like this in the lower Triassic, in the lowest Triassic. And even more astonishing, and this is some sort of derived archosaur, is this track. You can barely see it. Can you guys see the footprint in there? See, there's the middle toe, one toe heel, the other toe right up here. It's a nice 3-toe track. It would look like a smaller nithopod. No idea who made it. But a derived archosaur running around on the surface. So the tracks are telling us about the vertebrates, the macro megafauna that was running around at the time. Thanks to folks like Bill, we got some nice body fossils from the Triassic, from the Permian, mainly from the Triassic. But now the tracks tell us exactly where they lived and sort of what they were doing and why they were there perhaps. Maybe feeding on the things that were feeding on the invertebrates, you know, the preservation of the ecosystem. So what's going on in the soils? Well, there's lots of great evidence of traces in the soils. And these are burrows, vertebrate burrows, small ones. These aren't even the smallest. We have actually a smaller set where the diameter is like 2 to 3 millimeters, and they're helical. These things are about 2 to 3 centimeters. These are 5 to 6 centimeters in diameter. You see how these scratch patterns? We'll just walk through some of these so they're bigger. You can see them. Nice chevron-shaped scratch patterns, perhaps made by the claws or the teeth of these things. This is the top of the burrow. We've collected 100 of these things. This is the bottom of the burrow, so they have this sort of upside-down U shape, and I'll show you that in the cross-section. But you can see the scratches along the base here, and you can actually see the claw marks. In some of them, you can actually see where the hand just stops. You can see where the fingers come together. All right, so this thing would have had a sprawl. So some people call it a W shape. I call it an upside-down U. All right, and some of the work of Christian Sidor and others have said, yeah, these, some of the larger ones represent some sort of Cyanomathus or Elistrosaurus, and just take a look at stuff that Bill had in the museum, and they show me a little skull of Myosaurus. That could be, basically be the size of that burrow. That could be the possible trace maker of these things. And then you go the next size up, and we get things that are basically 10, 15 centimeters in diameter. So this burrow came down, we've excavated this and had it back in Kansas, came down, there's actually spiral here, and then comes down through the bed and terminates below the surface. And you can see this is all bioturbated. It went through the sand here, and there's actually, you can see the entrance part here, and actually made a little bit of a mound of sediment. So we actually were able to have the top of it preserved. So it looks like the opening of like a badger burrow. Maybe these things are more friendlier than badgers. No, it isn't against anything from Wisconsin, it's just the badger. The badgers just aren't, you know, really approachable animals. Even if you have candy, they don't like candy. And so this is the side view. Here's the top view You can see the same sorts of scratch patterns made by this. And this is the terminus of the burrow. And there's that lens cap for scale. So this thing is at its widest bit is at about 14 centimeters. It's possibly made by Cyanognathes. And then here's another one that does the exact same sort of thing. It comes down, and it's harder to see in this view. The other one I think will show up better. Comes down, curves, comes down underneath, and it keeps going. So this thing's over 2.5 meters, and we didn't have the very end of it. And there's from the side view. So it comes down from up here, curves around, and here's a lower bit, and it continues and goes into the outcrop. So it's probably also made by Cyanognathes. Then we get the big ones. And these things are huge. They're about 50 to 60 centimeters in diameter. And the same sort of pattern, this sort of upside down U shape. It's got the scratch patterns made by the feet of the excavator. There's the lens cap. There's a big ice pick for scale. Ice hammer, and then here's an ice, you can see the scratches. There's a terminal chamber, nice rounded, and then this thing continues up and goes up into the outcrop. So these things are gargantuan, perhaps made by Listersaurus. So you have a variety of burrows, but no body fossils, unfortunately. So we got to figure out, who did it. Well, you might say, so what? What does that tell us? Well, in the bigger scheme of things, what does that say? Well, they're probably constructed by these several varieties of different sizes of cynodonts and dicynodonts. They were probably used year-round, and like modern mammals, even though these things were mammals, they're therapsids, mammal-like reptiles. They're used for dwelling, brood rearing, maybe even hibernation, especially during the 4 or 5 months of darkness, perhaps. But this also tells us, because people are like, probably some permafrost, was pretty cold, but you don't see this kind of biorturbatio where you have permafrost. So it tells us that there's a lack of permafrost. Overall, the winters were much, much warmer. All right, so all this evidence converges to a pole that was much warmer. So you get extensive permafrost today at like 55 degrees north latitude. So this stuff will be warmer, much warmer, further south again. So when we put it all together and we do our diagrams of distribution of the burrowing and trace-making organisms, we have some of our aquatic traces we don't see again, but this could be preservational, maybe not from extinction. We don't see our anabranching or carbonaceous roots or self-looping, but we don't have those environments. These environments are those swampy, kind of lakey things. And we have roots and root halos right at the boundary. So there isn't a dead zone with no plants, but we don't have carbonaceous roots. We got those vertical burrows, therapsid tracks. We have the line here because we thought we found what looked like a nice track at Lamping Peak, but we're still not sure. Once you bring it back and look at it, it's just like, it could just be where some fish poop impacted it and looked like a footprint, and then the poop is gone, and so we just still working on that. But we got tracks, all of a sudden lots of tracks and burrows. Why? This stuff is well drained floodplains, much different condition. Our arthropod burrows, but what goes through? They're millipede and centipede trackways, annelid trails, the horseshoe crab trackways, the other fish swimming trails, Undicna. So behavioral wise, we don't see a big change. We just see a shift in the environment. So there are some things that don't go through, but it could be environmental control. All right, so to summarize all that, if you look at it, it's more of a hydroclimate shift. Your poorly drained landscapes, abundant ground cover with plants, and this time it's based on tree stumps and shallow roots and carbonaceous roots. Maybe a few footprints or not, that's a preservational thing. Abundant and diverse aquatic fauna and flora. We see even it looks like some biolaminates in the lake sediments. Forests with recurrent forest fires. And it looks like an increasing signal up section. Across the boundary, we still have abundant and diverse aquatic fauna, and even more things that we didn't see down here in the Buckley. Then we have abundant vertebrate burrows, and they're within 1 meter of the boundary, found at Colsack Bluff. So there is no dead zone of meters. They're right there, right over the boundary. And the floodplain sediments are much more better drained. They're still rapidly accumulating, but better drained. We have, of course, the body fossils that Bill and colleagues have collected, but now we get tons of footprints of these things and more. And then we know that the landscapes were well drained, but we know that there's abundant ground cover because of the roots. And fossil forests that the Taylors have found and working on. All right, so we can summarize all this. Basically, there's a hydroclimate shift across that boundary. Went from high to low soil moisture and water table conditions, improved the drainage. So it changes what's preserved, so it's a preservational bias. The Buckley, high water tables, shallow horizontal roots, diverse surface trails, poorly to well-developed histic paleosols, it's basically coals, peats that become coals, shallow roots, seasonal fires. Fremont, it's well-drained, poorly to well-developed soils, simple to complex branch roots that penetrate actually to 50 centimeters in depth. Lots of nice redoxomorphic features, so there's actually fluctuating water table, but in general it's pretty much better drained. Burrows, we have a small diameter to all the way up to the mega diameter, and some of the deepest penetrating ones go down 100 centimeters. Didn't have those things below, so it definitely reflects a hydroclimate shift rather than something else going on. And when we look at mass extinction at lower latitude to high latitude, it looks like it wasn't as severe at the higher latitudes based on our trace fossils as proxies for the organisms and for the hydrology and for the ecologic interactions. And we don't see any gaps, there's no dead zone. All right, and then lastly, there's abundant terrestrial and freshwater traces in the Fremont. And this is one of these indicators. When you compare them to the Triassic units that were deposited about the same time and later, in the mid to late Triassic, you got the same sorts of traces, the same diversity of things. So what we see at the high latitude is pretty much similar to what we see at the low latitude. So the ecodiversity, there really is no kind of difference. All right, there's some, but it's not great. Some people have hypothesized that the diversity of traces was higher at the poles compared to the low latitudes. So it really isn't. And we don't see a difference in a lot of these things, so they were globally distributed across Pangea. So that's telling us a lot about the climates. They're much more equitable, much more warmer. So we didn't have these strong latitudinal gradients. So allow things to expand more poleward. So with that, I thank you for your patience and your time, and hopefully this is food for thought in terms of, you know, why we need to look at past climates. I'm not saying we should go burn every forest and jack up the CO2, but we need to be educated and need to understand that, yes, there's always climate change, and yes, we can have some effect, but the world has been a warmer place. And it's also been a colder place. And if you look at the geologic record, it's actually been the Bahamas most of the time, not Fairbanks. So with that, I think.
So what does this teach us about the Permian-Triassic extinction elsewhere?
It's a good question. So elsewhere, so perhaps in the marine realm, maybe the biggest problems that they originated from the release of methane clathrates or things like that affected more of the marine aquatic ecosystems and forced to collapse that way. But on land, I mean, we lost things like the Glossopteris forest, but we still forest. There are other things there. So there was an extinction, but it wasn't probably as severe on land, but also at the poles where you're used to being in darkness 5 months out of the year and cooler temperatures. You know, the severity of that extinction event, now we now know is very complex, 2 pulses over 10 million years. If you're up at the high latitudes, it's no problem. They're used to that. You know, so Yeah, it's bad, maybe we lost 50% or 40%, rather than the 95% for the marine system and the marine collapse. So it's telling us it's not just a very simple pattern, it's very complex depending where you are latitudinally, depending where you are in the environment that you're in the sub-environment, the basin that you may be in. And this is also at the time when you're really pushing together the rest of Pangea, so whatever ocean floor was there, or shallows were going to push it up, you're changing the whole system, changing the whole oceanic circulation patterns, making one ocean. So of course you're going to have a bigger extinction pattern, an eventual tipping point in marine systems. Whereas the continental realm is like, great, we don't have to swim anywhere, we can walk. We can go to Antarctica, to northern North America, go to Canada. Well, back there's no Canada, but you know what I mean, right? You can just walk there, right? You don't have to, there's no oceans to clear, there's no nothing. So they behaved a different way and reacted a different way. And all we need to know is this, we actually need to go back and look more closely at the resolution of these things. Because if you look at the marine record, there are folks publishing papers now like, hey, we're actually finding all these trace fossils of things that should be in waters that should have no life. But you go to what should be a little bit deeper water and it's full of traces. The bottom is full of evidence of organism activity. So something's going on. And the pattern isn't as clear as we think it is.
Yeah, in the Permian though, it seems pretty clear to me that Antarctica is not a good place for land vertebrates.
It probably, well, that's the...
So when you look at land faunas, I mean, there's so many Permian rocks there that ought to have vertebrates in them. There's none there.
Well, that's the whole...
And so I think there's something in the terrestrial environment from the Permian to the Triassic that allowed vertebrates to go to higher latitudes.
Yeah.
That maybe wasn't quite there because of maybe the lingering effects of the glaciations from the lake.
There could be black glaciations, it could be the swampiness, and the other things we were talking with Christian out there when we were out at Graphite Peak, we just wondered, could it have been, maybe that they were there, they just weren't preserved. The reason why they may not have been preserved, it's all swampy and, it's slightly acidic. With cold swamps and cold water, you might not get them. What's interesting though, we were talking earlier, you go to Shackleton, the Permian-Triassic boundary is actually in the rock facies that represents the Fremont. So, and actually some of those facies, pardon?
Appears to be.
Yeah, appears to be.
According to Jim Collins.
Yeah, I think they may be right.
He might be right, yeah.
Because we don't see the big sands, but we see the same facies in which I think Christian or Roger thought they found an amphibian. Like if this is amphibian, this is below this boundary and it's in what we think is Buckley, but my colleague and I, Pete Flagg, doing the sedimentology, it's like these green rocks are just like the green rocks above. So the boundary, part of it could actually be in the Fremont, so that thing would be in the Buckley, but the Fremont facies stuff. So the time period, it would be Permian, but it'd be in rocks that represents better drain conditions. So I think it's a lot more complicated, but I think you're right, it has something to do with if it's really wet, they can't get there, or it's really wet and nasty, so they're not burrowing, you're not really preserving them, or they're not the environments that they really like, so you just sort of don't see them. Yes?
I have two questions. One, are we certain as to what caused the Permian extinction yet at this point? And the second thing is, we know that modern day coelacanths, who we have visited in the ocean, do not swim like fish. They swim like quadrupeds. Does the fossil record show whether that quadruped gait was found in these trace fossils or not?
Those are two very good questions. People still arguing, and Bill can reflect a little bit on this, people still arguing about the causes of the extinction, but we know better now is the two pulses, the rapid warming, and even more warming. tied to this big spike in the CO2, so it probably is methane clathrate, so some people do want to have, people do want to have meteorite impact, but there's no real evidence of meteorite impact causing that. So it's a big rapid warming and two-pulse warming, and with a major increase, really rapid increase in CO2, rapid increase in temperature, but also with the reposition of the, again, consider perhaps a tipping point, the change of the ocean currents and the reconfiguration of the continents. So I think people are coming to sort of consensus on that, though there's still arguments, because it's like, well, that's a lot of methane clathrate release, and would that be enough to do it? But that means release the methane. Methane's a good greenhouse gas, but then it actually becomes CO2 and it hits the atmosphere. And so, there's still a bit of debate, but we're learning more about that. And then in terms of the gait of the coelacanth and trying to watch it in the oceans, see how it swims. It's really interesting. These things were coelacanths that would have been remained in the freshwater systems probably since the Devonian. So their movement and patterns of the feet, we still, of the limbs, we still don't know that well because we don't have that well of preserved traces. But it's really interesting. You can actually see pattern that doesn't look like a walking gait or a crawling gait. When you get some of these things out on land, they sort of don't crawl. Sometimes they hop. The lungfish do the same thing, sort of the swimming approach, or they try to do a gait, but it comes out with the wiggling of their body. So it's that still have a long way to go to figure out, how similar it is to the modern coelacanth. So there's probably multiple kinds of locomotor patterns based on their, evolutionary involvement in freshwater and mixed freshwater terrestrial versus the ones that were out in the ocean and stayed out there. Hopefully that answers your question.
Where's the Morrison horseshoe crab?
Pardon?
Where's the Morrison horseshoe crab?
We got Morrison horseshoe crabs from Hanksville, from Colorado National Monument. It's a really good one. And it's been a while. There's like 2 other places. It's in my Morrison papers. Thank God I wrote it down because I got it down my head. So there's actually a number of occurrences, but the best stuff
In saltwash or?
in the Tidwell, saltwash, and there's a possible one in the Brushy Basin. It's just preserved pretty crappy. Oh, Cleveland Lloyd, where the Cleveland Lloyd coys are. There's some horseshoe crab traces there, and after that we just don't see anymore. And those are all definite freshwater. Some people still want to use those, and this is the problem with this, just using comparative anatomy alone. They're like, if it's horseshoe crab, it means it's marine. Because we don't see freshwater horseshoe crabs. Yeah, but they were, because there's no other marine evidence. Look at all, with all the vertebrates we found in the Fremont, we don't have any marine vertebrates. We don't have marine inverts. We don't have any polynomorphs that are marine. So the same thing in the Chinle. It's full of terrestrial and freshwater critters. Don't have a single marine critter except for this horseshoe crab. So it's the most parsimonious explanation. One really rapid marine incursion, and horseshoe crabs. That's like, us hanging out here. We go outside like, oh wow, salmon and starfish, and go to bed next day. Oh, okay, it was just what we ate. Or sea level fell.
Well, we used to have freshwater sharks.
Yeah. Those little things don't count.
Oh, okay.
But what's the most parsimonious thing? That these things evolved into fresh water, lived there for a while. And what's really interesting is if you look at the rock record, what happens in North America, right after the deposition of the Morrison, we get into some Cedar Mesa and some Burrow Canyon, and then you get a bit more, and then you get some there's a big unconformity, so something going on there, and then we start getting the western interior, so there's some sort of climate change, some sort of climate shift that's going on, and it gets even perhaps more humid, wetter, but we've lost the horseshoe crabs. They go away. So something has happened. Don't know, but they're gone. But things happen. You know, there's that famous shirt.
Guano happens.
There you go.
So, I can't remember if you mentioned this, but you've got all these tree stumps, but have you seen any leaf fossils?
Yeah.
Okay, because you haven't, I mean, obviously, you're not as interested in them, but I would assume you would have to find them in some of the same sediments.
Yeah, and actually around some of those stumps, beautiful Glossopteris leaves.
Okay, and have you seen any insect traces
Yes.
in the leaves?
Yes.
Okay.
And they're not like the really cool feeding patterns that you see in the modern ones. They're a lot more, or in the ones that you see later on, like in the Cretaceous and Eocene, particularly the Paleocene, Eocene. But they're much more simple. There are a couple of gulls, so there are gull-forming insects in there as well. And it's really hard to tease out. In the leaf litter, then we get things that look like, oh, they're in the leaf, but it's actually things going between the leaves. So you get all these annelids and worms, and perhaps earthworms. And that's the other big question. How is earthworms on every continent? How do they get there? People think, oh, based on their fossil, they evolved in the Oligocene. So then people say, well, big hurricane. That's a big Same thing with bats. How do you get bats across?
They burrow through the core.
Huh?
They burrow all the way through.
Yeah, that's right. Yeah, well, that's right. If Hollywood can do it, so, but, so you get these groups that are probably, pre-Carboniferous evolution, and then with the formation of Pangea, get them together, get them across the continents, and split them up. This probably was the same thing with the termites, the ants, and bees, and the wasps. People want to evolve all at the Cretaceous and the super radiation. But if you take a look at the beetles, and the beetles have a super radiation, but you can see their start at basically you get a really beetle-like thing in the earliest Permian, and then you get into the mid or late Permian, and it's a full-on scarabaeid beetle. It's in the Scarabaeidae. And that's almost looks like the thing that's from the early Permian. And then you have some insects from the Carboniferous or Pennsylvania, and then you get this big gap, and you get a few weird things in the Devonian, except for things like Columbella, springtails from Gilboa, New York. They look just like modern springtails. So there's a lot of gaps in the fossil record, and we're sort of misinterpreting them all as like, well, there wasn't anything there are a couple things, and then a couple more things, and then we get this big radiation of everything in the Cretaceous because it's the Cretaceous.
Especially beetles, because there are more of those than anything else.
Exactly. God loved
in the
those
world.
things. More, yeah, and
More beetles than anything else in the world.
was it over 30,000 species, or was it over 300,000? Some god-awful number.
Okay. Out of the arthropods that dominate anyway, the beetles have the biggest percentage by far. All the other invertebrates have 10%. The vertebrates have 5%. The arthropods have 85%.
85%. Can't ignore them. Allison.
I have one more quick question. So when you are prospecting for these trace fossils, what is your methodology? Because, I mean, obviously you get trained to see them, because most of those things I would just walk right over at this point. But are you just like...
I do.
That's
Because I'm not looking.
But, I mean, are you just picking a spot and cracking it open and hoping you find something?
No,
or?
it's just basically, it's like being out with Roger Smith, saying, walk around with Roger's like, oh, look at the skull, where?
Well, yeah.
It's like, oh my God, I can't.
But I mean, a lot of these seem to be hidden between layers,
Yeah,
so
so
you're not looking for something on the surface. So how do you know?
So you start, the good thing about these outcrops, right, they show the different surfaces. So you start looking and you find a good one, and you don't even have to keep cracking it back. I just start, okay, this is good, we document this, we collect these things, and then we can chase it back if we think there's something even more important or more of that what we want to get through there. But it's just, sort of, Bill was saying, I've got my eye trained for this. So I've gone to places with great scientists, like with Vitalik or Steve Dreesy or even Molly Miller, no traces. No, there's hardly any traces. When I was out there with Bill this time, I'm like, Man, if I don't find anything, NSF is going to roast me, because they gave me lots of money, and I'm gonna say, yeah, you're right, there's nothing out here. And I was afraid, I thought, I'm not gonna see anything. Like, we couldn't walk 5 feet without stepping on stuff. It's just, but my search pattern is for those things, and I work with the modern ones, so you start, you know, trying to figure out what they mean, but look at the modern traces, go excavate modern nests, and look at the facies pattern, the sediment pattern, so it helps, you know, understand what those are. So there's even stuff I collected I didn't see too much at the time, because I got photographs of them in the field, and they're in the lab. I had them sitting on the table in the low sunlight on the 3rd floor. I'm like, oh my God, that thing is hammered, just the whole surface, all these winding trails. I didn't see that. I saw some other crap on the other side, and you know, it looked okay, but that thing on the surface is phenomenal. So there's lots of things in there. So it's just sort of what you're trained to look for. I go out with these guys, I can't find a single bone. They're like, oh, look at this here, look at this
It's our
here.
methodology, it's just a different, you're looking for a different thing. He was in the lab today finding trace fossils in the
Yeah.
stuff that were thrown out.
Beautiful
Because
roots.
we're not paying any attention to the roots, we don't care about the roots.
Yeah, so
We're
this,
looking at the bone.
yeah, the Cryolophosaurus is in the soil. It's fantastic rhizoliths, and they're yellow tendrils. So, well, that's telling us that preservation of the roots, that it was basically sort of pretty poorly drained. So they're basically gurtide, laminite, run some XRD on those things, but really poorly drained. So they're preserved as yellow tendrils. It's just basically iron roots, and some of them have a little bit of manganese. The thing about that is it's poor. That's another indicator of being poorly drained. So there could be spherites, really tiny things. So is it an iron siderite, something else? Right, so the indicators are really poor drainage, and they're really fine, thin roots. You get a little masses here, and then disseminated. But you know, he's showing me how great work prepping that thing out and picking up a couple scraps. Like, have you guys seen any traces? No, I don't see any traces. Take a look at some of this stuff. Hey, look at the roots! You guys are pitching this?
Final messages, don't throw anything away.
Yeah, and then, yeah, you don't want to throw it away, or no, it's even, so, well, that's the other big message, you know, that you can't get anyone, particularly Washington DC, to understand is, just because you went somewhere doesn't mean you know it all. Just because somebody from some big place said, well, I was there and I didn't see much, you gotta keep looking and go back. New eyes, new search patterns, new devices, new paradigms. Paradigm shifts all the time. And you just can't say, oh, well, we already did that. You know, we need to do something else. You know, and one of the things people say that we know, they say we know the surface of Mars better than we know the surface of the ocean. Well, guess what? We know the surface of the ocean better than we know what the hell lives in the soil, on land. We're putting up malls and parking lots, and we're whacking all these arthropods and annelids and other things that are in the ground. We don't even know. We don't care. Ah, looks it's okay. There's more dirt over there. Go play there. So I mean, just as bad, we don't know what's going on in the subsurface. What's going on in our beta zone and our phreatic zone. So, you know, the critical zone is so important. All these zones are important. We just got to keep going back, keep funding science, keep funding research, and fund it the old-fashioned way. It's like, let's go see. It's a fishing expedition. We don't have to go and test something specific. You know, and to go and look like the explorers. But instead of being on the Beagle, we can be in the Traverse. Go and look.
You guys don't get that. That's what I've been driving around with.
Very nice.
So everything that you were finding in this study, over how many seasons were you there looking at?
That was one season?
That was one season?
Yeah.
Wow.
7,000 pounds of rocks. I am not a very popular person at the University of Kansas. So where do you put all these things? It's not my problem.
Not all of that.
This is science. We're going to find a place to put them. And then maybe perhaps what we don't need will become part of the rock
You
art.
probably brought back 5,000 or 6,000.
You guys brought a whole bunch with those dinosaurs in them. That's okay. It's like, what's all these? This looks like hieroglyphics. What are you, an archaeologist? No, I'm an ichnologist, so this is important. But seventh, yeah, one season. If you gave us 2 seasons, oh my God.